Growth media is used in microbiology to grow microorganisms that require nutrients to synthesize four critical macromolecules: proteins, carbohydrates, lipids, and nucleic acids. We will be using three different powdered growth medias and water to create selective and differential media to obtain pure cultures of specific bacteria. We will first measure out the correct amount of powdered in grams for each growth media for 100 milliliters of water. 100 milliliters of added water will create enough growth media for about 3 petri dishes of selective media or differential media. I then added the 100 milliliters of distilled water into a glass beaker and placed it over a hot plate and dropped in a magnetic stirring bar. Once the hot plate is turned on and stirring, I added the powdered media to the beaker and allowed the mixture to come to a boil. Once I add the powder to the water, you can see that each media has a different color due to the different ingredients needed for the selectiveness and differentiation between microbes. After the solutions have come to a boil, I'll allow the mixtures to cool for a few minutes before transferring them into 3 separate Erlenmeyer flasks. If the solutions are left to rest for too long, the media will set in the beaker and begin to gelatinize. If this happens, you may have to heat them back up on a hot plate before you transfer the solutions. I then added aluminum foil to the tops of the flasks to cover them. We are now ready to place them into our autoclave. I do not have a proper autoclave at the moment, so this pressure cooker will have to do. Ideally, you want to sterilize the solutions in an autoclave that can heat up to 121 degrees Celsius and build a pressure of 15 psi in order to ensure proper sterilization. Once sterilized, we allow the solution to cool for a few minutes and we can now transfer the media to sterile petri dishes. You want to fill the petri dish 1/2 to 2/3 full. Here are a few different types of media that I have used to inoculate bacteria. These are old plates that have overgrown, but you can still see the growth of bacteria on the plates. Media is great for determining types of bacterial infections in clinical settings and is still used today in medical laboratories.